Today, I'd like to share fascinating insights from a recent study titled Human Skin Triglycerides Prevent Bedbug Arrestment. This study explored the interaction between bedbugs and human skin compounds. Bedbugs have become a global nuisance, notorious for their resilience and the difficulty they pose in indoor pest control. While they're attracted to humans for feeding, they intriguingly avoid staying on the host afterwards, preferring instead to hide out nearby. The researchers hypothesized that while certain odors from human skin attract hungry bedbugs, other compounds might actively discourage them from lingering or resting on us after feeding. To test this, they designed a series of experiments aimed at identifying these compounds and understanding their effects on human bedbug behavior. The researcher methodology involved 5 different stages. First up is collecting the bedbug specimens. The study used 4 different bedbug populations, 1 laboratory-reared strain, and 3 collected from infested apartments. All bedbugs were maintained under controlled laboratory conditions and were fed rabbit blood to standardize their physiological state. Next up was the human skin swab collection. 6 human volunteers of various backgrounds participated. Volunteers followed a strict protocol to minimize external variables. They avoided spicy foods, showered without applying deodorants or cosmetics, and refrained from strenuous activities before sampling. Then, skin swabs were collected by rubbing filter paper on specific body areas, arms, legs, and armpits, to gather natural skin compounds. After skin swabs were collected, then the bedbugs were set up with choices. The core of the study involved setting up controlled arenas where individual bedbugs were given a choice between 2 shelters: a control shelter, which was an untreated filter paper tent, and a treated shelter, which was a filter paper tent conditioned with human skin swabs or extracts. Bedbugs were introduced into the arena during their active phase and allowed to move freely overnight. The next morning, researchers recorded under which shelter the bedbugs chose to settle. After the researchers saw which shelters the bedbugs were choosing, they then did an extraction of skin compounds phase. To pinpoint the active compounds, skin swabs were extracted using hexane, which dissolves nonpolar substances like oils. The extracts were then fractionated using solid phase extraction, SPE, to separate them based on chemical properties. Each fraction was tested separately in bioassays to determine its effect on bedbug arrestment. Lastly, they did a chemical analysis of the skin compounds. Behaviorally active fractions were analyzed using advanced techniques: thin layer chromatography to identify lipid classes present, gas chromatography, mass spectrometry for detailed chemical profiling of volatile compounds, and lastly, liquid chromatography, mass spectrometry, to analyze non-volatile, larger molecules like triglycerides. Now it's time to analyze the results. Across all bedbug populations and human volunteers, bedbugs consistently avoided shelters treated with human skin swabs. This avoidance was significant regardless of the bedbug's origin or the volunteer's age, gender, or ethnicity. When shelters were treated with hexane extracts of skin swabs, bedbugs showed a dose-dependent avoidance. Higher concentrations of the extract led to a stronger preference for the control shelter. And lastly, the bioassay-guided fractionation revealed that certain fractions containing non-volatile lipids were responsible for the avoidance behavior. Chemical analysis identified triglycerides, tags, as the primary compounds in these active fractions. These triglycerides are common components of human skin oils, otherwise known as sebum. They are produced by the sebaceous glands. Lastly, researchers tested various commercially available triglycerides by applying them to the shelters. Bedbugs universally avoided the shelters treated with different types of triglycerides, regardless of their specific chemical structures. This suggests that the tags in general signal bedbugs not to stop and rest on treated surfaces. So concluding this study, the study provides evidence that human skins emit multifunctional chemical signals. First up, they attract bedbugs. Volatile compounds from our skin help bedbugs locate us for feeding, but at the same time, they deter bedbugs. Non-volatile compounds like triglycerides discourage bedbugs from staying on us afterwards. So why do bedbugs perform this behavior? Well, researchers concluded that this behavior likely benefits bedbugs by reducing the risk of being detected or removed by the host. It also helps bedbugs return to safe hiding spots where they can digest their meal and reproduce. So, what are the potential applications of this new knowledge? Understanding the role of triglycerides opens avenues for new bedbug control strategies. By incorporating tags into materials or surfaces, we might prevent bedbugs from settling in certain areas, which in turn will disrupt their life cycle. So overall, this study sheds light on the complex chemical interactions between humans and bedbugs. It highlights how compounds naturally present on our skin can influence bedbug behavior, balancing their need to feed with the necessity to avoid prolonged contact with the host. Such insights are crucial as we seek innovative solutions to manage and control bedbug infestations. If you like and subscribe to our channel, we'll be sure to go over more research articles in the near future.